Hello everyone, my name is Stephanie Yang. I am a senior at the Math, Science, and Technology Center at Paul Laurence Dunbar High School, and today I'll be telling you a little bit about my research project, which is the role of corticosterone in house-sparrow responses to novelty. It was conducted under the mentorship of Dr. David Westneat at the University of Kentucky. To give you a brief overview of what I'll be discussing throughout this presentation, first, I'll start with some background information about my project. Then I'll move on to my purpose and methodology before presenting my data, results, and discussion. Then I'll wrap it all up with my acknowledgements and works cited. To understand what this research is about, you will need to know that many organisms live in an environment that's constantly changing. Due to these circumstances, there are various sources of uncertainty that may reduce in an organism's ability to predict the outcomes of their decision. For instance, a bird might find a nice sturdy tree perfect for building a nest in, but little does it know that this tree was scheduled to be cut down in a week. After spending much time and effort on its home, all of it goes to waste, putting the bird in a very disadvantaged situation. Unfamiliar or novel stimuli in particular tend to be perceived as threatening, as the organism cannot be certain if the stimulus poses a risk or how even best to respond to it. It is believed that an organism's reaction to such stimuli is related to its stress response system. The stress response system of a bird is largely mediated through its hypothalamo-pituitary adrenal axis. The activation of this axis causes the major glucocorticoid hormone in birds, corticosterone, also known as cort, to be secreted from the adrenal gland. In this experiment, house sparrows were used as a model system to investigate how cort levels affect animals' behavioral responses to novel stimuli, which will expand our behavioral understanding of both house sparrows and other organisms that live in similar conditions. The purpose of the experiment was to find out how would house sparrows respond to novelty and how will artificially elevating their cort levels affect this response. I predicted that the house sparrows would, with higher cort levels, will be less likely to feed because higher cort levels stimulates elevated stress, which may be expected to result in higher caution. Here you see a visual representation of my methodology. So for my methods, 11 house sparrows were tested. Before I could conduct any trials, however, the house sparrows had to learn certain behaviors to reach their food, which was placed in a feeding board. As shown here, these are feeding boards, which are just boards with 9 wells, where the outer 8 wells are filled with sand and seeds, and the middle well is usually kept empty, except during trials. First, I will need to allow the subject to get used to its new environment, which is an aviary with 4 platforms where feeding boards are placed. I'll scatter a generous amount of seeds on top of the feeding boards, shown in this figure here. So the house sparrows know that it's going to find food there. After they know where to find food, I'll place 9 seeds in each well. The sparrows were taught how to dig for their food when I slowly bury the 9 seeds deeper and deeper into the sand after each feeding. So after each feeding, if I see signs that the sparrows were picking at the sand, making it fly everywhere, and the shell of the seeds are broken apart, then I'll know I can bury the seeds deeper, but if not, I'll keep them at the same level until it finally learns that it needs to dig for its seed. After you learn how to dig for their seed, I'll teach them how to flip their lids. When I place these lids on the outer rims of the board, I'll slowly move them in towards the wells. After, then eventually it'll cover the wells. This is all done because I'm trying to make the sparrows work for their food as they probably would in their natural environment. After all these behaviors are learned, I needed a way for house sparrows to take in court. So I would feed the house sparrows an artificial diet, which is basically just a white substance powdery mix. It's a protein powder to which court could be added to. This was done by giving them a seed to diet mixture. The ratio of seed to diet started out to be mostly seed before slowly increasing the diet after each feeding goes by. After about 2 weeks or so, prepping the subjects, they are finally ready to begin their trials. The novel stimuli used was a large, brightly colored building block. There was 6 object present trials, 3 with court diet and 3 with control diet, and 8 object absent trials, 4 with court diet and 4 with control diet. 10 minutes prior to each trial, the artificial diet was taken out on each feeding board, so the sparrows were given time to feed and digest the diet. Then when the 10 minutes were up, the outer 8 wells, and depending on the type of trial, so if it was a object present trial, a building block would be placed on the board. The artificial diet was left on the board during the trial because its effects wear off in about 30 minutes. So to make up for that, it's left in the boards. The color of the building blocks change with each trial, so the sparrows wouldn't get used to the stimuli. They were then observed through a camera for about an hour, where their approach latency, which is how long it took them to approach the feeding board, and whether or not they fed from the feeding board, were recorded. Here you see 2 pictures. It's just a visual representation of what an aviary would look like. So here are the forward platforms where the feeding boards go. Here you can see a sparrow feeding from artificial diet in the middle well. And here you can see a sparrow approaching a feeding board with during a novel object present trial. Here you can see all the novel objects on the edge of its board. So for my data and results, in this graph here, it shows the likelihood of house sparrows to eat from the feeding board, where the green bars represent the control diet and the brown bars represent the court diet. The y-axis is the proportion of trials in which the subject fed, and the x-axis is whether the novel object is absent or present. When the novel object is absent, the subjects fed from the feeding board during all trials, so 100% of the time. And when the novel object was present, on average, subjects feed from the feeding board in less than half of the trials. So half of the trials would be 50% of the trials, and the majority of them don't feed. And in the second graph here, it shows the approach latency in seconds, which is measured on the y-axis. Of the house sparrow to the feeding board. When the novel object was absent, except for a few outliers shown here, most of the subjects approached the feeding board in less than 500 seconds. But when the novel object was present, majority of the subjects took anywhere between 1,200 seconds to 4,000 seconds to approach the feeding board. Looking at these 2 graphs, the court diet does not seem to be having much of an effect on the sparrows, as it follows the trend set by the control diet. So I thought that if they had an elevated court level, that they would be less likely to feed from the feeding board. But looking at this graph, it is very similar to the control diet, so not much of a difference. To test for the significance of the novel object and the diet, I used a 2-way ANOVA test, which compared the means of the approach latency grouped by novel object presence, diet, and the interaction between the 2. The only thing we need to look at here is the p-value column. So we're looking for a p-value that's less than 0.05, because if it is, that means it's significant. So the core elevation, which is the diet, does not significantly affect approach latency because it was found to have a p-value of 0.6116660, which is higher than 0.05. But the novel presence did significantly affect approach latency since it has a p-value that's less than even 0.001. So what does this all mean? Well, the results did not support my hypothesis, which was that house sparrows with higher court levels will be less likely to feed from feeding boards. The novel object presence had a significant effect on house sparrow approach latency, as expected, but the court diet ended up not having any significant effect on approach latency. I thought this may be because the novel object had already stressed the house sparrow out to the max, to the point where spiked court levels will have no effect on their responses. It is also possible that the court diet did not induce a large enough court spike to affect their behavior. Because of these conclusions, for further studies, it would be interesting to maybe use smaller novel objects next time so it's less stressful on the sparrows, or maybe even larger doses of court. I would like to wrap this all up by thanking my mentor, Dr. David Westmeat, and a PhD student, Allison, for helping me out throughout this whole process. I would like to thank the University of Kentucky, the Kentucky Young Researchers Program, and the Math, Science, and Technology Center for allowing me to participate in such interesting research, and my friends and family for always supporting me and giving me rides to lab. Here is my work cited. If you have any questions about this experiment, feel free to contact me at my email here. And thank you for tuning in.